Extending a warm welcome. Our topic today is on a perspective overview. If you are new here, we are a group of retired professors behind Team BioTech Whisperer, channel sharing videos in life sciences. Let's dive into the topic for today. Examining the functions of microbial genes is crucial for understanding the ecological roles and intricate genetic interactions that occur within microbial communities. Despite the growing popularity of microbiome research, a lack of practical in situ editing techniques hinders experimental design and advancement, making genetic understanding and manipulation of microbial communities largely unattainable. This is known as the CRISPR conundrum. CRISPR-Cas systems are used by bacteria as a form of adaptive immunity against threats such as viruses. Scientists have adapted these systems to cut out or replace specific genetic sequences in various organisms. In a recent study, researchers from North Carolina State University demonstrate that viruses engineered with a CRISPR-Cas system can overcome bacterial defenses and make precise changes to a targeted bacterium even when other bacteria are nearby. The study's authors used two different engineered bacteriophages to deliver CRISPR-Cas payloads for targeted editing of E. coli, first in a test tube and then in a synthetic soil environment that mimics real soil, which is known for harboring various types of bacteria. The researchers found that both engineered bacteriophages, T7 and lambda, successfully targeted and delivered payloads to the E. coli host on the lab bench. These payloads expressed bacterial fluorescent genes and altered the bacterium's resistance to an antibiotic. The virus used in this case targets E. coli by delivering DNA to it in a similar way as if it were injected through a syringe. The study highlights the potential of using viruses as a tool for targeted genetic manipulation of bacteria even in complex environments. The researchers then used the lambda bacteriophage to deliver a cytosine base editor to the E. coli host. The base editor functions as a kind of programmable on-off switch for genes in E. coli. Rather than CRISPR's sometimes harsh cutting of DNA sequences, this base editor changed only one letter of E. coli's DNA, demonstrating the precision and sensitivity of the system. These changes deactivated certain bacterial genes without making any other changes to E. coli. With a system like this, scientists can make highly precise single-letter changes to the genome without the double-strand DNA breakage that is commonly associated with CRISPR-Cas targeting. To test the system in a more realistic environment, the researchers use a fabricated ecosystem loaded with a synthetic soil medium of sand and quartz, along with liquid, to mimic a soil environment. They also included 3 different types of bacteria to test if the bacteriophage could specifically locate E. coli within the system. The study shows good efficiency in finding E. coli and making the targeted genetic changes. This further supports the future application of this technology towards discovering the genetic basis of key bacterial interactions with plants and other microbes within highly controlled laboratory environments such as fabricated ecosystems. The study represents a significant step forward in the use of CRISPR delivery by utilizing viruses to deliver CRISPR machinery in a complex environment. This can be seen as a mechanism to aid the microbiome. With the ability to make targeted changes to a specific bacterium while leaving the rest of the microbiome untouched, this proof of concept has the potential to be applied to any complex microbial community, leading to potential improvements in plant health and gastrointestinal tract health, which are important for food and overall health. The researchers plan to continue testing the phage CRISPR technique with other soil-associated bacteria. This study highlights the potential to manipulate soil microbial communities to control the composition and function of bacteria associated with plants in fabricated ecosystems, which can be used to understand how to enhance plant growth and promote plant health. This is of broad interest for sustainable agriculture. The combination of a synthetic microbial community, soil matrix, and EcoFAB device in this study offers a controlled and reproducible model that more closely mimics the process of in situ editing of the soil microbiome in real-world conditions. Closing perspectives. The CRISPR-Cas system has had a transformative impact on agricultural biology by serving as a powerful functional genomics tool. It has greatly advanced plant breeding and crop improvement by speeding up the development of improved cultivars, increasing genetic diversity, and helping in the domestication of wild and orphan crops. The field of gene editing is rapidly evolving, with various advancements such as the development of different Cas effectors with broader target range, increased efficiency, and the ability to make more precise DNA modifications through base editing and prime editing. The current collection of various CRISPR reagents allows for gene knockouts, targeted gene insertions, precise base substitutions, and multiplexing.